everyone, today we're going to be looking at surface area to volume ratio, which can also be written like this, SA to V, surface area to volume ratio. As usual, make your notes as you watch this video, as well as using the textbook to make your notes. Pause the video, rewatch parts as you go, and write down any questions that you have and bring them to class. So the study design dot point for today is surface area to volume ratio as an important factor in the limitations of cell size and the need for internal compartments organelles with specific cellular functions. So we've looked at this second part here, internal compartments and organelles, and now we're going to look at the surface area to volume ratio and how it is an important factor relating to cell size. There is a video linked here or linked in the PowerPoint slides if you've downloaded them that is useful, and I highly recommend that you watch that video. So how do you calculate surface area to volume ratio? Well, first you're going to do the surface area, which is length times width times the number of sides. Okay, sorry about my writing. I'm writing this on my computer. So if we take this cube over here, what we're going to have is a length of 1 times a width of 1 times 6, because there's 6 faces or 6 sides, and we end up with 6 centimeters squared, which we can see here. Then you're going to calculate the volume. So volume is length times width times height. And for this cube here, the length, width, and height are all 1 centimeter again. So we 1 times 1 times 1, which is then 1 centimeter cubed, which we can see here. So then you're going to write it as a ratio. So you're going to take the surface area, which is 6, to the volume, which is 1, and you've got 6 to 1. So with a cube that has a side length of 1 centimeter, you end up with a surface area to volume ratio of 6 to 1. As you can see here, there is a larger surface area compared to the volume, which is what we want in cells. The larger a cell gets, the smaller the surface area to volume ratio. What this means is that the greater the surface area and smaller volume, or high surface area to volume ratio, can help a cell exchange materials efficiently and effectively with the environment. So when we have a much larger surface area compared to the volume, it will allow cells to take in nutrients relatively fast and efficiently, as well as excrete wastes relatively fast and efficiently. If we've got a really big cell, it's going to take much longer for nutrients to get in, as well as waste to get out. I think this image really sums up the importance of having a high surface area to volume ratio, because if we were just made up of one gigantic cell like this, we can see that there aren't that many entry points for nutrients or gases like oxygen to get in, and it's going to take a very long time for those nutrients and gases to get all the way into the center here. However, we are made up of multiple cells like over here, and there are multiple entry points into each cell, and it will take a much less time for the nutrients or the gases to get all the way into the cell. So having a higher surface area actually means that nutrients, gases, or waste can move in or move out relatively quickly, and our cells can get the nutrients, gases, or whatever they need fast and effectively. So why are cells so small? As we saw on the previous slide, it allows us to have much more efficient transport of nutrients and removal of wastes. So if we look at a human over here, a human is made up of about 37 trillion cells. So an animal cell is about 50 micrometers long, which is about half the width of your hair. So if the width of your hair is about 100 micrometers, half of that is what an animal cell is. So a human, an adult human, is made up of 37 trillion cells. And as we saw in the last slide, it is much more efficient to transport nutrients, remove waste if we have multiple entry points and we have a high surface area compared to the volume. If we were just one large cell, it would be highly inefficient to transport nutrients in and out and remove wastes. And that wouldn't aid survival. What does aid survival is being made up of trillions of cells that have multiple entry points that increase our surface area compared to our volume. So the shape of cells or structures in organisms has an effect or impacts the efficiency of transporting nutrients and waste in and out. For example, flattening a shape increases its surface area. These are red blood cells. They have a concave disc. This is because they don't have a nucleus. So there is larger surface area compared to the volume. Small flat cells with projections and compartmentalized cells are more effective at nutrient transfer than large spherical cells. And we know that from previous slides where more entry points equals faster entry or exchange of nutrients and wastes. So as we can see here, if we had a cell that was really long and flat, it has a much larger surface area compared to the volume than a cell that is just a cube, which has a surface area of 3:1. This one is 4.25:1. We can see this in multiple cells or structures in our bodies. So this is intestinal tissue, which has these projections. This means there is more surface area, so more entry points into your intestines so that nutrients can be absorbed by your body. This is the same for microvilli in alveolar cells, so in our lungs. Because of these little projections, there are much more entry points, so there's a higher surface area compared to volume, which allows us to take in as much oxygen as possible because there are multiple more entry points than if it was just flat. If it was just flat, there'd be way less entry points than there are because there are multiple villi standing up like this. Here's a close-up look of an epithelial cell that has microvilli. As you can see, this microvilli vastly or greatly increases the surface area compared to the volume, and we're able to take in the nutrients or the gases or whatever it is we need much faster and more effectively because we've got a high surface area compared to volume. This is very obvious is in our gastrointestinal tract. As we know, it's really curved. So we have lots of different curves in our gastrointestinal tract. Inside the intestine, there's also villi, which are these projections. And on top of the villi, we have these cells. And you can see there is microvilli on top of the epithelial cells. So we are creating lots of folds, lots of projections to increase the surface area compared to the volume. So we increase the rate of nutrients coming in or waste going out. We can also see this in much larger structures. So if we look at this elephant over here, as we know, elephants live in very hot areas, and an elephant is huge, so it's going to retain a lot of heat. One of the ways it allows heat to be released from its body relatively quickly is through having very large, flat ears. So an elephant's ears has a high surface area to volume ratio, which allows heat to be released really quickly from their ears. On the other hand, if we look at this walrus, which you can see is very round, very large. A walrus lives in a very cold, icy environment, so it needs to keep as much heat in as possible. So it has a low surface area to volume ratio because there is a high amount of volume compared to the surface area. So low surface area compared to volume, because it wants to take a long time for heat to travel all the way from in here outward. So having a high surface area to volume ratio benefits cells particularly, but also structures in animals like elephants releasing heat from their ears. And having a low surface area to volume ratio benefits animals that live in cold environments because they want to retain heat. They don't want to lose heat very quickly. So I hope that helps you understand the importance of surface area to volume ratio at a cellular level, but also at a whole organism level. Please bring any questions that you have to class, and I will see you then.